All right.
All right, so you can start if you like. For the students, well, we are going to start with the lecture 7, so sorry for the delay, and well, you can start with your lecture if you like, Marcus. Thank you very much.
Yes, and then we have to see whether I can get to the second part of it. All right, so sorry for the delay. I'm going to talk about the structure and diversity of and life cycles of viruses today, and I'm working at the Oceanographic Laboratory in Villefranche. What I see here is we have a change in names that I didn't do. It's still Paris, but it's the Sorbonne University, and our institute is not called anymore Observatoire Océanographique, but IMEF, which means the Institute for the Sea. So, slide 2. So I give you a little introduction on the history of viral community ecology. Already in 1979, by using transmission electron microscopy, a high viral abundance was found on 0.2 micrometer filters, although most viruses are smaller of that. And also in 1980, that was a freshwater systems, high viral abundances were detected by electron microscopy. But this kind of information was ignored, probably because people were at that time more interested in bacteria and to show that marine bacteria are very important. But then, about 10 years later, when it was very, very well established that marine bacteria or microorganisms in general are very crucial, high viral abundances were found in aquatic systems by Berger et al. And Nature paper, and the year afterwards it was also shown that the infection rates of phytoplankton and bacteria plankton were very high. So then it was time to go into this part of research in terms of microbiology. So what is a virus? A virus is a heterogeneous and non-phylogenetic or non-monophyletic group of biological entities, and the genome can consist either of DNA or RNA and is encapsulated in a protein code, in a protein code that is called a capsid. So now I'll show you some pictures of viruses. These are bacteriophages. You know, you see all the spikes here. Can you see my cursor? No? Okay. So they have different morphologies. Also here shown different sizes of viruses in a wetland system. And then we have lemon-shaped viruses on top, or viruses with spindles and things like that. And so they have also some morphology. Okay, this is a sample from a natural system where a bacterium on the lower part is surrounded by viruses. The same here, so this is probably getting totally infected in a few seconds. Then we have here a bacterium that is completely filled with viruses. The same here. On top, we have a long filamentous structure, and on the right you see, hopefully you can see that it is completely full of viruses. And I counted that there were about 500. The lower three pictures show a filamentous bacterium that is in the process of dividing, and all of the cells are infected. And in the lower part, there are several bacteria that are completely infected with viruses. Okay, so now we go to the part, how do we enumerate viruses? And okay, so what is usually done is to take a water sample, and this water sample is diluted, and then you spread it along with bacteria on a plug that's called then the plug forming units. And so when you see when there are viruses and they have a specific host of the bacteria, then they make holes, that's shown on the lower left part. And then you can count it and calculate back to the abundances. That's the old traditional way, but now we have also ways to study the variosphere on the community level. And to the left you see cybergreen staining and epifluorescence microscopy. The larger dots are the prokaryotes and the lower ones are the viruses. So what you do here is you stain the DNA in the viruses and in the prokaryotes. You filter the sample on a specific filter and use epifluorescence microscopy. On the right side you have the same thing using transmission electron microscopy, where you spin down the samples on a grid. But that's way more time consuming. So what this data also, all these pictures also show you already, that Virion plankton is very roughly said about 10 times more abundant than bacteria plankton. This method are being slowly replaced by, are we in the right slide? Yes, by flow cytometry. And so you have the same staining with cybergreen or similar DNA stains, and then you do a detections in the flow cytometry, and you can also, as you see on the left side, get different groups of viruses. You can at the same time also count bacteria. The other thing is you can still use transmission electron microscopy, because a very nice thing is that you can see when viruses are inside bacterial cells, and that helps to determine a very important parameter, and that's the burst size. And the burst size means the number of viruses that come out of a bacterial cell when the bacterial cell lyses. And here you see a couple of samples where the bacteria are completely full with viruses that fill the maximum burst size, or other samples where they start getting built up. It's called the minimum burst size. And that's what you need to estimate virus mortality. And I'll show you that later. So the distribution of viruses is about like that. On the left, we have the biomass of the three important groups of microorganisms in the ocean. That's the prokaryotes, which means bacteria and archaea, then the protists, and then the viruses. So biomass of protists and viruses is approximately similar and much lower than that of prokaryotes. But the abundance of viruses is much higher than the two others together. This is a distribution of viruses in different depth layers of the ocean. That is the epipelagic, that goes down to 200 meters, the mesopelagic, that goes down from 200 to about 1,000. Then we have the bathypelagic area, the deep sea, that goes down to 4,000 meters. And then the abyssopelagic, that is even going further down. And all the dots you see here, most of the cruises, or data from most of the cruises that have been done so far. So here we go to the samples where we compare the ratio of the abundance of viruses to the abundance of prokaryotes, and it's only shown for the depths of 100 meters and lower. And you see this ratio is not extremely different. And to show that in another way is that the VPR, the virus to prokaryote ratio, is in coastal waters about 20, and in the deep sea it's 28, so even slightly higher. And that's here shown in another sample from a cruise of Africa. That in surface waters we had about like in 100 meters a virus to prokaryote ratio of 10. But in 3,500 to 4,000 meters, we had a prokaryote ratio of 100. And that's very strange, because prokaryotes decrease in this depth layer by at least an order of magnitude, but viruses do not decrease much. And that's sort of an enigma, because how come that the viral abundance is so high in the dark ocean, although the host abundance is only increasing by 1 order of magnitude? One potential reason could be, and I come back to that later, is vertical and lateral virus transport. Another one is prophage induction. Prophages are viruses that sit into the genome of the host, and they can get out of it once in a while. I'll come back to that back later today. But what is then the inducing agent for that? What I think personally is more important is that in the deep sea, it's very cold, so there's low temperature, and there is, because there are only a few bacteria, the enzymatic activity is low, so there are potentially low degradation of viruses. And it also could be that we underestimated due to the problems with pressure, because you have, in the deep sea you have a high pressure. When you bring up the samples and measure virus production, maybe we do some miscalculations here. Okay. So in the ocean, there are about 10 to the 30 to 10 to the 31 viruses. If you assume about 50 nanometers of a viral capsid and you align that, you could make a viral pearl necklace that's wrapped 100 to 1,000 times about our galaxy, the Milky Way. Okay, so what I have to try now is to go to the second part. Of my talk, and I don't know how to find that. Oh my goodness. How do I go back here? Okay, I think that works now. Okay, so second part. Viruses on particles. Virus-particle interactions. This is just a few pictures. To the upper left, we have again what I have shown you before with epifluorescence microscopy. The larger green dots are DNA-stained bacteria. The very small ones are the viruses. What you see in a little bit of bluish lectins that stain exopolymers of bacteria, and this is then called the organic aggregate or marine snow particle. To the left there is the same thing, but done in thin sections. And the lower part is again a similar samples with all the green parts DNA stained. Here, if you see that the lower right part, it's a diatom, and all the other reddish parts are cyanobacteria. That's the three, again, cybergreen staining of the nucleic acids. The small dots are the viruses, the bigger ones the bacteria. And in red, that's a lectin that's used to stain the exopolymeric substances such as cell walls of bacteria and bacterial capsules. And this is a 3D volume reconstruction using laser scanning microscopy. Here we have laser scanning microscopy of two other types of particles. The upper one is a soot particle, or black carbon particle. The lower one is a Sahara dust particle. So what you do here is you make scans of sections and then do a reconstruction of the particles and the bacteria attached to them. Now we come to the mortality of viruses, also called the viral decay. So when a virus absorbs the cell, it's gone from the environment, but it could still be in the cell. Viruses can be absorbed to particles and inactivated, or remain, or are removed from the system by thinning out. High temperatures are not good for viruses, and that's very good for COVID this year. Enzymes killing viruses, they are ingested by viruses, and what I did not mention here, and that's also that solar radiation, especially UV radiation, is detrimental to the DNA of viruses, and that's hopefully also going to happen for the SARS viruses this summer. Okay, so how do we estimate virus production in the ocean? There are basically four methods right now. The one is to look at the visibly infected cells. I have shown you pictures before that. Then we can estimate the decay rates, and assuming in a steady state that the decay rate has to be replaced or has to be equal to the virus production rate. Then we have tracer approaches, and then we have an approach that's called virus dilution or virus reduction approach, and that's the one that's used most currently. So the visibly infected cells can be done by thin sectioning of bacteria. I have not shown you a picture of that, or the whole cell approach. That's the many pictures I've shown you before with viruses within the cells. I don't think I have one here, no. All right. Oops. If you have this, like the percentage of cells with a virus in it, a mature virus, you have to use conversion factors, because for a large part of the infection cycle or the latent period, viruses cannot be seen. And then from using viral production can be used from the frequency of infected cells, which includes also the part where you cannot see viruses, the burst size. I've talked to you about that before. That means the number of viruses that are released when a cell lyses, and bacterial production. So then you can do decay rates. That is, you take a sample and you inactivate the viruses, and you count the decay of viruses over time. The other one is you add a specific virus into the plaque-forming unit assay to see the changes. And as the biologic activity is stopped by chemical inhibitor, such as cyanide, the decay over time equals in a steady state the virus production. This can be done by PFU, I don't think I've shown you that in, okay, let's stick with the PSU, forget about the other one. The MPN is for phytoplankton. Or you can do total counts. The tracer approaches are that you use radioactively labeled thymidine and study the incorporation rate of that into viruses. And another one is that you use fluorescently labeled viruses as tracer and add that to seawater. So the radiotracer approach, viruses samples are incubated for some time. Then you separate bacteria and viruses by filtration and measure the amount of viruses that is incorporated into viruses, and then using conversion vectors to calculate that the number of viruses that have been produced. This method has not been used very often because it's very complicated. The fluorescence-enabled viruses are easier to study because you take a water sample, you concentrate the viruses, you stain the viruses, then you add them to water samples. In the second sample, you count the total amount of viruses, and then from the virus production and the decay rates, you can calculate the change of the virus's abundances given decay and production, and in total then estimate the total production. And then you have dilution approaches where you reduce the virus abundance in the sample by rinsing out the viruses, or you add virus and bacterial plankton concentrate to virus-free water, and then you follow virus abundance over time, and this is the production. You can also do a stepwise dilution where virus is a dilution with virus-free water and with virus-rich, and then from the differences in virus abundance over time, that's a bit complicated to calculate. You can also estimate virus production. That's called the virus dilution approach. The other one is called the virus reduction approach. So how do you do these kind of things? So you take a water sample, you remove using a 0.08 micrometer filter in offshore waters. In coastal waters, you use 1 micrometer filters. So you remove traces and larger particles. Then using a 0.2 micrometer tangential flow filtration, you can concentrate the bacteria, make a bacterial or prokaryotic concentrate. And with the filtrate, using other cartridges with like 100 kilodalton, you can concentrate the viruses if you want, but you can also make the virus-free water. And then if you take the bacterial concentrates and put it into virus-free water, you can do this virus reduction approach. And you can also do a lot of experiments with that, with and without viruses, but I think that's what comes tomorrow. So and what happens then here is you look at in these approaches with bacterial concentrate and virus-free water, you count the viruses over time, and these viruses that are produced have to come from bacteria that have already been infected. So you, with the virus-free water, you inhibit new infection. So all that you find and an increase of viruses comes from already infected cells. At the same time, you can also do decay studies to compensate for that. So we have here, like the green line, it would be this increase in lytic viral production. But then, as I show you right in the next slide, there is also the stage where viruses can remain within the host genome. That's called lysogeny. And you can add mitomycin C, which induces the lytic cycle for those. That's here shown in the red line. So the difference between those would be prophage induction. That's a very important viral life strategy that you can study in the same way, so at the same time. All right, so we have here the viral life strategies. For you, concentrate on, or for the classes, concentrate on the lytic and lysogenic or temperate ones, but they will also take you through the other ones. So you have a cell, that's the cell wall in red, and in black the GDOP. So the viruses attach to the cell wall and inject the nucleic acid. Then you have several ways how viruses can behave. Viruses can, for instance, that's the chronic infection, replicate a little bit, not too strong, but get viruses out of the cell once in a while without destroying them. That's the chronic infection. Then you have, that's one of the major ones that we know, where the viruses take over the host genome, so it's also called the hijackers. then they produce first DNA, then the protein pack the DNA into the protein, then lyse the cell, and all the viruses get out and the cycle comes up again. But there's also the possibility that the DNA of specific viruses, not all of them, but specific viruses, the temperate ones or lysogenic ones, that this DNA gets integrated into the host DNA. And then the cell divides and the virus prophage divides with them. And that's called the lysogenic cycle. That's the hitchhikers. At some point, it can, however, happen, for example, when there is a DNA damage, that the virus decides to leave the host cells and then they enter the lysogenic, the lytic cycle, okay, and also kill the cell. And something in between that's not very well understood is the cytolysogenic cycles, where a little bit of viruses are produced as well. Okay. That's an interesting picture because this is a bacterial cell with a capsule around it, and on the lower part you see that a virus is going through this capsule because the virus has to get to the cell wall to inject the DNA. So what viruses can also do without having ATP, they can depolymerize, not digest, but depolymerize the capsule of bacteria to get to the cell wall. Now a little bit to the relationship between lytic stage infection and percent lysogeny from a study I did a long time ago. And what seems to be here is like what we have in the dark ocean, that's on the left side, that lysogeny is more important. That is, that the viruses or that stay as prophages within the host genome, and that the fraction of visibly infected cells is more important in coastal systems and also in anoxic bottom systems. And if you look at this relationship, there could be a tipping point here shown in green, where systems switch from one type of viral life strategy to the other one. And that's data from a station in the Mediterranean Sea to show this kind of thing that I showed before. What I talked to before, we had like the frequency of infected cell was going down from the epipelagic area from 14 to 4 to the bathypelagic area, whereas the frequency of lysogenic cells was increasing from about 12 to 70%, 12% in the epipelagic area to 70% in the bathypelagic area. It's also interesting that the size did not decrease with depth. Also, there are other studies showing that the lytic viral production decreases with depth, whereas the induced viral production, which means when you add mitomycin C, then you induce the lytic cycle in the lysogenic cells, remains roughly stable. This suggests that lysogeny becomes more important into the dark ocean. Well, this one I've decided to skip. So there's this theory that lysogeny dominates in environments where there is low growth of the host, like in the deep sea. So this is compatible with the idea that lysogeny is an adaptation to environments where either the hosts are difficult to find due to the low abundance, or because viral abundance is decaying rapidly. There's also evidence that the prophages can repress unnecessary gene expression and help hosts in non-favorable environments. In this sense, lysogeny would be kind of such a mutualism or symbiosis. So it's that prophages are rather a key to the host survival in disease than dangerous molecular time bombs. So what would be the inducing agents in nature? Inducing would be that the prophage is getting out of the host genome and then enters the lytic cycle and kills the cell. We know that sunlight or DNA damage is a cause for that, so that's leaving the sinking ship. Other factor is in the same phase hydrogen peroxide that also starts to be built up in the ocean when there's a lot of UV light. Another completely different way is that when there's a very fast growth of hosts, then the viruses also come out, because then they have a lot of chance to infect the new other cells. And that's the party time scenario. And also a funny thing is that sunscreen can induce the prophages in the ocean. So now a little bit about viral diversity. So we have different types of viruses that probably developed during evolution independently. So we have double-stranded DNA viruses. They have like a helix. We have single-stranded DNA viruses, which have only wired up DNA. Then we have single-stranded viruses that come from the 3' side, and others from the negative sense from the 5' side. Then we have double-stranded RNA viruses, single-stranded viruses, and also again positive sense and negative sense RNA viruses. They very likely developed independently. And we have also different types of viruses. In the sense, we have bacterial viruses, that is bacteriophages. We have viruses from archaea. We have eukaryotic viruses. And we have also viruses that infect other viruses. That's the virophages. And there is little overlap between those viruses. So we have, in terms of isolated, we have not the bacterial viruses that can infect other viruses or other cells, or vice versa. This on the left side, this is a community fingerprint called rapid PCR, which is a very rapid one, and this is the depth profile from 5 meters to 2,000 meters. And just by looking at this gel, you see that some bands, like here, are only found in between 600 to 2,000 meters. There's a band that's found only in 2,000 meters, and then there are bands that are only found in surface waters. So clearly, viral diversity is different along depth profiles. And this metagenomic part, if you do the DNA analysis with different primers, give you a kind of different but overall similar results. But what I want to show you here actually is that this black part is sequences that are completely unidentifiable. That is, they have no match with databases from isolated viruses, and this ranges from 70 to more than 80 or 90%. So this clearly shows that there's a huge part of viral sequences where we have no idea what it means. This is certainly something for future times to be analyzed. At least to try to do that. So this is data from a study that has been conducted with the Tara Ocean System from the Arctic to the Antarctic, and what they found was that there were mainly 4 clusters of viruses. And here in yellow, that's the surface water or epipelagic viruses, which are separated here in like this reddish thing from the mesopelagic viruses. Mesopelagic is between 200 and 1000 meter. And then here on top in violet, there are the viruses from the deep sea that's below 1000 meter. And then we have another cluster, and that's here in blue and gray, and that combines viruses from the Arctic and from the Antarctic. So they can't be further away than that, but obviously the cold temperature and the special conditions promote the growth of similar type of hosts and similar type of viruses. All right. Again, to the depth distribution of viruses, we have in the photic zones specific viruses that you don't find anywhere else, or only in very, very small quantities. Then we have viruses that's called here ubiquitous viruses. You find them, and also the host bacteria. You find them in surface water, and you find them in the deep sea. Reasons for that could be sinking mechanisms. That is where viruses get attached to organic aggregates or to black carbon that is soot or to Saharan dust or to volcano ash and sink out into the deep sea. Some of the bacteria can survive that. So viruses can also be, or virus host systems can also be transported into the deep sea. Viruses and bacteria alone, they're too small to sink. They have to be attached to organic aggregates. Or there's another possibility. Then there's physical transport when like a water current hits a continental plate and the water is, because it's, for example, cold, it's going down, and this can also transport viruses into the deep sea. And then it seems also to be that some of the viruses are just endemic along with the bacteria to the deep sea, because we know that there are bacteria that are very well adapted to the special conditions into the deep sea, for instance, the high pressure that is down there. Okay. So from these studies on metagenomics, in this paper I've shown you from pole to pole, there were about 200,000 DNA-specific viruses found. And again, as I said before, the majority of the sequences has no match in databases, so there could be even more. And this study did not it just involved double-stranded DNA viruses and not all the other ones. But there is evidence that RNA viruses are almost as diverse as the DNA viruses. So overall, diversity of viruses in the ocean is huge. To look at that in another way, as far as we know, that for every bacterial and archaeal and eukaryotic species, there is at least one specific virus. In many, there are several specific viruses. So this means that, or this is another way to look at it, that the diversity of viruses should be higher than the species diversity of cellular life, in terms of species only, not in terms of functions. Okay, and that's the finish of the talk for today. Begonia, can you hear me?
Yes, yes, I'm here. So, wait a moment. Yes. I'm here, so if you students have any question, this is the moment to do them.
I can see you now.
Yes, here I am. So, have you got any question about this talk? Gloria? Yes?
Yeah, hello.
Okay.
I wanted to ask if is there any record of that virus could infect humans?
I did not understand. I couldn't hear it.
Yeah, with the virus that you find, like that they are living in the ocean, is it possible that they can infect humans or not?
Well, usually not. But what can happen is that, you know, so if there are not good sewage systems, you can have viruses getting into the human viruses getting into the sea. And that's a potential problem, especially in areas where there's a lot of where the water is not very clean, where you should not swim. For a specific thing, like as we have this COVID problem now, I think COVID does not survive very long in seawater because the COVID virus is very special. It has a lipid bilayer around the protein capsid where you have these spike proteins, you know, that's are the ones which are used for vaccinations. And if you put such a lipid into the seawater, it usually disrupts. So the fear that has been there that SARS-2 could infect on the beach or in seawater is not there. But there is a theoretical point that at least in very polluted water, that you could also get virus infections, but only from the viruses that have been produced by viruses and then transported into the sea. There is no singular case known that like a native marine viruses has infected a human person.
Okay, thank you.
Okay. Melina, now you have a question?
Yes, good morning. Okay, I have two questions. On the first presentation, you mentioned about the enigma of the viral abundance in the dark ocean compared to the abundance of its host. And one of the reasons you mentioned was vertical and lateral virus transport. Is it related to the physical properties of the sea and how we can transport viruses from layers, or is it because of another reason?
Okay. I'm having a certain dispute with people about that because a lot of people think that a lot of viruses are transported along organic aggregates or volcano ash or Saharan dust into the deep sea. Because viruses themselves, they cannot sink. They're too small. Right? Bacteria can also not sink themselves. They're too small. So they have to get attached. But then if you look, there also, we know about, this can happen. So that's clearly the case. But I think it's in most environments, it's not quantitatively important. Or in some maybe, but in others not, because we also know from areas where there is not a lot of sinking particles, where there is a lot of lateral transport, like in the center of the Pacific Ocean, we still find these high viral abundances in the deep sea. So while it can be in form of diversity, for example, important, but in terms of quantity, this sinking, I think, is not very important. I guess it has rather to do with, as I said, with the low temperatures in the ocean, in the deep ocean, because that's between 1 and 4 degrees. And so the degradation rates of the protein is low. There are not a lot of bacteria down there, and they're not super active. So the enzymes will degrade that. And what I realized, and what we should do for next year, I make a note here, is there's also the theory, or not only the theory, but we know now also in the deep ocean, it seems that bacteria and archaea are creating large particles. They are also called the dragon particles. And it could be that when bacteria are archaea are concentrated on these large particles, they have again a high contact rate with the viruses. And so you could have a high production down there. I'm not sure whether I talk about that tomorrow, but I made a note that then for the lectures, if you have that next year, that this will be included. Thank you for these questions. But you said you have a second one.
Yeah, thank you for your answer. I also was looking at the graph on the second presentation about the change in the lifestyle, and I just wanted to confirm, like, the tipping point that you mentioned, is it a tipping point because you stop having dots from the Mediterranean Sea and you only start seeing dots from Baltic Sea and Gulf of Mexico? Is it because of that?
Well, that's a good question. I don't think so, because there are also other data showing that. I think it's rather related to the host abundance. So if you have not a lot of hosts, it's better to stay within the genome, because if you lyse the host, you will not find another one. And in other systems, it's different. And I also have to say, it's this relationship is only true if you look at very large-scale data. In different systems, it can switch in a very different way. So I didn't want to go too much into details of that, because it's getting then extremely complicated. But this is just to show that there are potentially environments that favor one of the lifestyles. Other people have found that such things can also happen on a seasonal scale, so that in winter, when it's cold, there's more lysogeny than in summer, when there is a higher temperature and more organic matter available. Does that answer your question?
Yeah, I can understand the complexity of this situation.
I didn't want to make it too complicated. Right?
Thank you.
Okay, now Roanna, please. Do you have any question? Roanna?
No, the mic is not working.
Sorry, my mic isn't working. There's a chat. Is there any work of fakes being used to treat domestic seaweed to reduce the load of pathogens? In the chat.
Well, there's a thing that, let me answer that in a different ways. There's a thing that's known for a long time, that's called phage therapy. And that was before you could use antibiotics. Phages were used to fight E. coli.
Mm-hmm.
So phages were isolated from E. coli. And then when people were sick, they were just taking them as medication to help them. And this works to a certain extent. And this has been picked up again for years now, because of the problem that the antibiotics are making this problem that bacteria get immune to them. just resistant. But they're not resistant against the viruses, and the viruses have the benefit you can manipulate them so they're not dangerous anymore, but they can still kill the pathogens. So that's an avenue that's getting more and more important.
Mm-hmm. Okay, thank you. Okay. Do you have more questions for Marcus? No? Well, we can finish with this lecture. So I think that there is an assignment or a quiz related with this lecture. We have you have some questions about the lecture, so they will have to answer them. And I think that it will be all about this lecture.
Yeah, and so, also the second lecture for tomorrow is uploaded, and also the quiz.
Okay. So no more questions, so we can finish with the lecture.
And there's also the, you know, if there is you can always come to, you have my email address, and if you want to know something, you can always write me an email if you, like, you know, like a question pops up today, tomorrow, and the next days. There's no problem.
Okay, they have your email in the introduction, so they can be in contact with you, of course.
Yeah, so don't hesitate to do that. There's no problem.
Okay. So we can finish with this lecture, and we'll go on tomorrow.
Yeah. Okay.
Thank you very much, Marcus. Tomorrow I will be connected again because maybe I don't think that tomorrow will have any problem, but I will be connected at 8:30.
I think now I know how to do that.
Okay. Okay, well, see you tomorrow, Marcus.
Okay, thank you very much.
Okay,
Goodbye.
thank you.